Welcome back to many of you, and thank you for joining us at EnviroLogix today for our third and final seminar on genetic editing in agriculture. My name is Jamie Welch. I'm a market segment leader at EnviroLogix, the global leader in rapid GMO and mycotoxin diagnostics. I'm pleased to introduce again our resident genetic editing expert, Dr. Emily Whiston, who will be completing her genetic editing seminar series. Emily has worked in the lab as an R&D molecular development scientist and has managed DNA-enabled product development teams at EnviroLogix. In 2018, Emily has been recognized as a subject matter expert in genetic editing, presenting at the Organic and Non-GMO Forum to large global grain companies and non-GMO certification bodies. And now I'm excited to pass the presentation over to Emily.
Thanks, Jamie. I'm excited to talk about genetic editing again in this third session, the biggest buzzword in science, with major implications for plants and ag as well as throughout biotech. So we talked about the basics of genetic editing and a little bit about the science of how it works, and now we're really going to talk about how scientists are putting it to work, what this means for us as a diagnostics company and throughout the ag industry. As a reminder, EnviroLogix makes rapid, point-of-need, mycotoxin and GMO diagnostics for the global ag supply chain. Our diagnostic products are applied in trait development and seed breeding, and then through the grain handling and processing space. Specifically, why am I talking about CRISPR as a representative of EnviroLogix? So my academic background with a PhD in microbiology focused on comparative genomics. So that was taking whole genomes from multiple organisms and representatives within those organisms and comparing millions of base pairs to look for patterns that give rise to certain traits. And it's this science that makes genetic editing possible. You can't edit what you don't understand, and comparative genomics and sequencing has basically paved the way for the major breakthroughs that genetic editing has been able to accomplish in plants, in animals, and bacteria, basically throughout biology. So in this third session, I'm going to give a quick recap on what is genetic editing and then talk about this new generation of ag traits. So what are the broad trends and impacts on ag? Why does this present a challenge for diagnostics, and how is EnviroLogix approaching these challenges? Genetic editing is basically a flexible new toolkit for genetic modification. So on the left side of the screen, these are sort of the simple tools in a toolbox prior to genetic editing, from traditional breeding and then mutagenesis, RNAi, and transgenics. So transgenics are what we typically think of as GMO, or genetically modified organisms. And what makes genetic editing so exciting is that it takes all of those tools and puts them into a single, relatively easy to implement, super versatile new biotechnology that can accomplish all of those prior engineering or approaches to gene modification, puts them into a single toolkit that can do all of those, plus more precisely and more rapidly. So some examples of genetic editing that I covered in the prior two sessions. So here are a couple of examples of gene knockouts. So specialty crops, this is a waxy corn created by the company Corteva. So this is actually, waxy corn is a native trait, but Corteva was able to use gene editing to recreate that native trait in a high yielding corn background. Another example is this non-browning mushroom. So knockouts can be used to slow ripening or reduce byproducts. So these mushrooms, a scientist used the knockout process to create a non-browning mushroom. So when those mushrooms are sliced, they'll stay fresh when packaged. Precision base editing. So precision base editing or precision breeding could be the rapid introduction of desired native traits or novel traits without associated yield drag. That's very common for crossbreeding. So an example here, and this is theoretical because this is actually a scientific article about the underlying genetics, not a gene editing application, although it could be used for gene editing. So this is a theoretical example where these scientists figured out that there's a native SNP, or a single nucleotide polymorphism, so one base pair change out of millions in rice that confers cold tolerance. So gene editing could be used to recreate that cold tolerance in a different variety of rice if that was beneficial to a farmer. Another example, this was an example of SISgenic sequence insertion, so taking DNA from one variety of an organism and putting that into a different variety of the same organism. So again, rapid introduction of desired native traits without yield drag. So here, scientists were able to take an ancient allele, or a native piece of sequence from beef cows that confers the polled, which is the hornless trait, and put that into elite line, high milk producing dairy cows, so that those dairy cows and their offspring would be born without horns, would not grow horns, and wouldn't have to go through the painful, costly process of dehorning. Another recap that I covered in the first session, so most genetic editing traits will not require USDA regulation. So this is actually the letter to the scientists who created that non-browning mushroom from the USDA. And the USDA said that a GE, or genetically engineered organism, is deemed a regulated article. So again, the USDA has regulatory oversight only if it has been genetically engineered using a donor organism, recipient organism, or vector that meets the definition of a plant pest. So plant pest would be DNA from fungi, viruses, or bacteria that cause plant disease. And the reason for this is that the USDA regulatory process is tied to the Plant Protection Act of the year 2000 that was specifically geared towards plant pests and preventing the spread of plant pests, so that genetic engineering wouldn't threaten sort of broader agriculture in the US. So because most genetic edited traits will not have plant pest DNA in the final product, the USDA has basically come out and said that it does not fall under their regulatory purview. So the broad outcome of this is that genetic editing will mean more molecular traits in more places. So if this is a new, very versatile bioengineering technique, it will lead to rapid precision breeding of diverse traits. And then if those have a non-regulated status through the USDA, that significantly reduces product development time and cost. And when I say significant, I mean tens of millions of dollars, by many estimates up to $100 million, out of the engineering development process. So what that means, reduced time and cost, opens the door to editing in more crops, more traits, and by more companies than we saw previously with sort of traditional transgenic approaches. And that is having major impacts on the trait development market. So despite major consolidations at most of the big ag companies, the Bayer Monsanto, the Dow DuPont merger, the Syngenta buyout, startups are actually making the overall ag trait development market more complex. So companies like Calyx, Cebus, Caribou, AgBiome, and Pairwise. And those startups are entering the market with new ideas and new types of traits. So here is the product pipeline from Calyx. And you can see they have sort of two major categories. They're calling consumer-centric products, like a high oleic or healthy oil soybean, to a high fiber, a reduced gluten wheat, a cold storage potato, reduced browning, and then some what they're calling farmer-centric products, so drought-tolerant soybeans, herbicide-tolerant soybeans, powdery mildew or plant path-resistant wheat, herbicide tolerance, higher quality alfalfa, etc. So a different mix of traits, including some very sort of consumer-focused types of traits, which is what we haven't really seen out of the transgenic or GMO product pipeline previously. Overall, this presents new challenges for diagnostics throughout the food chain. So if we think about what the current or transgenic GMO testing process looks like in the food chain, starting with seed breeding, seed development, and farming, through the sort of grain elevator and processing space, export, and then through to ethanol plants and millers. On the left side, so where grain is still whole grain and seed, the diagnostics there are largely very rapid, so 5 to 10 minute on-site immunoassays, which is EnviroLogix' primary specialty. And then once that grain has moved through the food chain and goes into a more processed material, that is typically sent out for a reference lab service, usually PCR, and that's pretty expensive testing that takes multiple days to get an answer back. And there are common challenges for both of these. So the major challenges would be access to sequence information. You can't detect what you don't know. And then the expansion of the number of traits and crops to be tested. On the protein side, a lot of these new traits will be non-protein traits, but current DNA testing is typically longer than 10 minutes, which doesn't work with how grain elevators and a lot of the breeding process is set up for very rapid detection to try to move trucks through and get bins and storage filled to go through to export. And then on the reference lab PCR side, a lot of those tests rely on common sequences. So the CMV35S promoter is a sequence that's been used in many GMOs. So where one single test can capture multiple types of GMO. Again, PCR and reference labs will be faced with a major expansion of the number of traits and crops that need to be tested, and that creates significant complexity for their business. So how is EnviroLogix approaching these challenges? So we continually monitor the market for emerging trends and traits. It's worth noting that there are molecular traits already in the market. So, but they're already stacked with traits that do express a transgenic protein. So that one of our relatively simple, rapid, low-cost immunoassay protein-based strips can capture GMO because it can go after the transgenic protein. So Plenish and Vistive Gold are high oleic soy varieties that include a transgene for Roundup or glyphosate resistance. So even though these are not gene-edited traits, they are molecular. This high oleic itself doesn't express a transgenic protein, but the glyphosate resistance is transgenic. And then we're also growing our technical capabilities with rapid molecular detection. So again, I mentioned that to this point, molecular detection takes longer than 10 minutes. So we are investing heavily in a technology called DENABLE, which is a rapid nucleic acid amplification method that generates results equivalent to PCR, but can work with crude sample extracts, and results are generated in 10 to 15 minutes, and we're working every day to make that quicker, faster, and easier. So how we develop assays as an example. So this is a feasibility study for the molecular trait Arctic apples. Arctic apples are an RNAi trait, and here is a relatively simplistic view of what that looks like. So DNA is transcribed to RNA, and RNA is translated to a protein. So here in Arctic apples, the scientists were looking at the pathway, the proteins involved in conventional apple bruising and browning. And what they were able to do with RNAi, so RNAi, they transcribe a short RNA which binds the target mRNA, and that creates what's called a RISC complex. The RISC complex is targeted by the cell for destruction, so that blocks the creation of the proteins involved in bruising and browning, which has made the trait the Arctic apple, which is a non-browning, non-bruising variety of apple. So in our feasibility study, we took a look at the genetic construct of that RNAi trait. So there's a promoter, what's called a leader sequence. There's the RNAi trait itself, and then there's the TNOS terminator, which tells the cell when to stop transcribing new RNA. So we looked at the TNOS terminator, and then we found an apple control sequence. So we went through an assay design where we looked for sort of ideal primers and probes to create an assay, screened dozens of assays to choose the best performing assay, put those 2 assays into a duplex so that in a single well or a single test, it would detect both the apple control sequence as well as the TNOS terminator. So got that duplex optimized, went through a sample prep development, and then put this assay for feasibility through specificity and accuracy testing. So here was the outcome of that. So the sample preparation here that we developed is under 15 minutes, so it can work with either an apple leaf or skin punch or a dried apple sample. Add a buffer, heat, centrifuge or spin it down, and then dilute. So that's the sample prep process, and then that crude sample can go directly into an assay. So our assays are lyophilized, which means that they are shelf stable with all of the components needed for a reaction. All it needs is sample. So the sample goes in, and then for this feasibility study, this was run on a sort of traditional qPCR thermocycler, but it could also be run on a less expensive piece of instrumentation. So again, that assay, getting the assay onto a plate and then running the assay, it was also less than 15 minutes. And then the outcome there, so for TNOS, which again would be the molecular component of the Arctic apple trait. So NTC is a non-target control, which is negative in both the TNOS assay as well as the apple control assay. Arctic apple, so these were dried apple, they're calling them app bits, and they're available from Amazon. So the app bits were positive for both the TNOS from the Arctic apple trait as well as the apple control assay. A conventional apple chip, so another sort of dried out variety of apple, but that did not contain the Arctic apple trait, was negative for TNOS, so negative for the Arctic apple trait, but positive for apple control. And then apple leaf, as well as a conventional apple from the grocery store, negative for TNOS, but positive for the apple control. And then we also ran a control using a 10% grind of a GMO corn, and that GMO corn did include a TNOS sequence. So that sample was 100% positive for TNOS, but negative for apple. So there was a TNOS positive, but it didn't come from an Arctic apple trait. It came from a different corn trait. So some key takeaways. Smaller companies and startups are entering the trait development market with new and different ideas, which is making sort of the overall ag diagnostics market pretty complex. So again, these wide-ranging new traits make diagnostics more complex from both a technology and assay development perspective, between sort of the wider variety of traits to be tested as well as access to sequence information. And how are we responding? So EnviroLogix is committed to best-in-class market knowledge and technology to meet the needs of our customers. And so this concludes our 3-part series. So I'll say again, farewell from Maine. And today I have a picture of a lobster boat, perhaps one of our, probably our most iconic bit from Maine. We hope sometime you'll come visit us and have a lobster. If you have any questions or would like to contact us about a custom assay development, I hope you'll email me or our sales team. So my email is ewhiston@envirologix.com. Thanks so much for listening.